So right after my internship, I got a very unique opportunity to participate in a research cruise with my lab. I actually gave the final presentation for my internship on the boat, which was very exciting. So the research cruise was dedicated to studying this gelatinous animal. So the animal at the top is called a siphonophore. These are very understudied, and we wanted to know how they fit into the food webs in the open ocean system outside of San Diego, so in the Southern California area. So I got to spend 2 weeks aboard Scripps research vessel called the Sally Ride, which is pictured below. And I got a lot of experience working with hands-on techniques and data collection. So I got to learn how to retrieve and deploy a lot of sampling nets, as well as other oceanographic equipment. I also learned how to identify a lot of deep sea animals, as well as sort and process the ones that came up in our nets. And most importantly, I think, is that I got the opportunity to make connections with a lot of people in science at varying stages in their careers. So I got to talk to grad students, postdocs, who are people who have gotten their PhDs but want more experience in research, as well as professors. And so this experience was really unique because almost everyone on the cruise was a grad student or older. So I was actually the youngest person on the boat. But that gave me a really great opportunity to talk to these people in an informal setting because we were basically living together for 2 weeks. Next slide. So of course I had to include some pictures. So because of COVID-19, there were a lot of restrictions and regulations we had to follow. One of those, just to make sure everyone was safe on the boat. And one of those was a 2-week isolation period before the cruise. 1 week in our homes, and then 1 week we had a hotel isolation. So they put us in a hotel room and we weren't allowed to leave. So the picture on the left is a picture of us at the hotel. Unfortunately, some of the members of the science party, our balconies were connected, so we got to go out there and talk to each other. Also because of COVID-19, there was restrictions as to how many people were allowed in each lab. So while on the boat, we were split into working groups. So the middle is a picture of the group I was in. We were operating the net that you see on the left. Upper right is a picture of me in front of 2 of SIO's other research vessels, and the picture on the bottom is a picture off the back deck. And this was kind of my view for the 2 weeks. We were out in the open ocean. Next slide. The right is a picture of a squid that I actually got to study this summer. It's called a strawberry squid or a cockeyed squid, and it's called that because it has one really big eye and one really little eye. So you might be able to see that in that picture, and that's an adaptation that it has for living in the deep sea. And in the middle is a video of comb jelly. So if you play that video right there again, it's kind of swimming around, and it has these rainbow. The light refracts off of these little appendages. It calls cilia. So hopefully you can see that there. And one really cool thing, so on the left, we see at the top is a picture of a CTD, which is a sampling equipment that we had. It measures physical characteristics of the ocean. And on cruises with CTDs, they have something called Cup Day. So in the bottom left, you can see we got to decorate these styrofoam cups. They attach these cups to a bag to the CTD, and they send the CTD down to depth. So these cups actually went to 2,000 meters depth, which is around 1.25 miles. And because the pressure is so great in the deep sea, the cups shrink. So you can see a before and after picture of the cups. So that was definitely something that was really interesting.